Thanks. So, yeah, so as I said, I'm in, actually, we're in the Department of Cancer Systems Imaging. So my lab is located down on the South Campus of MD Anderson. So you'll find that a lot of the basic research labs for MD Anderson are not, at least the newer, some of the newer ones are popping up down there. So if you guys could switch over to the next slide. There we go. So our lab has been interested largely in prostate cancer, which is one of the most common cancers in men. And if I could if you kind of migrate to the upper right there, sort of encapsulate sort of what I want our lab to do is hunt down new signaling pathways that we think drive, in particular, the later stages of the disease. And so the prostate cancer is sort of atypical in the fact that it's largely a steroid hormone driven cancer, driven largely by androgen, testosterone. And so a lot of the pathways that we hunt down are related to androgen signaling, but then also we find that a lot of metabolism as well. And within our department, we find that metabolism lends itself to a lot of imaging modalities that we can sort of leverage later on. From there, I sort of view us as we hunt down pathways or proteins that we think, for example, are important in the disease. I kind of think of there's two branches then. One is we do the hardcore validation in terms of knockdown, overexpression, see if we can drug certain pathways. That comes with any sort of identification and implication. The other is really a lot of our labs spends this time doing basic research in terms of, okay, we've identified some sort of molecule that we think is important. What regulates that? And so with the understanding that perhaps whatever we found might not be the best therapeutic target. And so maybe there's something upstream of it that could be better, and then also elucidating sort of how it works. And so this gives us ways to assess whether or not that pathway is going to be active in disease. I would expect anybody that comes into our lab, any graduate students, to kind of be at least familiar with different aspects. Some of the ways that we hunt down pathways is we leverage a lot of clinical data, either that's publicly available or some of our clinical collaborators in GU medical oncology at MD Anderson or throughout the country and world, and be able to analyze these with some sort of bioinformatics tools, whether or not it be very basic bioinformatics, like using a portal, or some people go on and learn how to code. That's obviously is a powerful approach. Then comes a lot of the mechanistic work in, for example, cell culture, where we'll do molecular genetic approaches of modulating different pathways. And then those things that seem like they're most promising, then we move forward in a variety of different animal models, whether it be xenografts or genetic mouse models or PDX models, what have you. Typically, as we move forward with certain pathways, then at some point, if we think that they can be a therapeutic target, then we'll work with different groups, either companies, to see whether or not what options there are. I would say right now there's probably, for graduate students that would come into the lab, I kind of view sort of two major projects of where I think would be good starting points. One is related to a kinase called CAMPK2, which we initially identified as having a major role sort of right within prostate cancer cells, as shown in the xenograph on the left, on the right-hand bottom side. But one of the things I think that was a bit surprising to us that we found recently is that there's clearly pro-cancer roles for this kinase outside of the cancer cell. And so in probably the immune system and also in the bone, which is of interest to us because prostate cancer mainly metastasizes to the bone. And so we think that elucidating some of these mechanisms might open up new therapeutic opportunities. And then probably one of the other major pathways is looking at this master regulator of cell metabolism called AMPK, which has had sort of this mysterious role in cancer, where some labs say it's pro-cancer, other labs say it's anti-cancer. We now have evidence to suggest that to date, people have sort of been oversimplifying its role. And we think that it's because that there's actually different subpopulations located throughout the cell. And depending on which subpopulations you activate and for how long, you get very different effects. And so these projects really need somebody with a strong training, I'd say, in biochemistry and cell biology sort of to tackle them. These are just starting points where I think there's a lot of tools and reagents, but certainly, as people become more and more familiar in the lab, there's freedom to roam, certainly scientifically. And I'm just sort of there as sort of a spirit guide, so to speak. So I think I'll stop there and take any questions if anybody has them.